It's my great honor and pleasure to co-chair with Professor Xiaobo Wang in this section. The first speaker in this section is Professor Yan Yun. Professor Yan got his MD from Taipei Medical College in year 1982 and has his PhD in pathology and cell biology, Thomas Jefferson University in year 1998. He is a very famous researcher in cancer, and he became the president Taipei Medical University in year 2011-1, and since year 2018, he is emeritus president, chair professor, and the director of Center for Cancer and Translational Research at Taipei Medical University. Because of his significant contribution in cancer research, he has got a lot of awards. If I name this award, I think time will be very limited. So let us welcome Professor Yan Yun. He will give us a speech entitled Recent Adventure in Cholangiocarcinoma, focus on molecular profile. Professor Yan, please.
Yeah, thanks, Professor Wu. Yeah, it's very hard to follow Ming Chi's talk, although I also couldn't get a hug as well. Yeah, so not just you. Yeah, and today I would like to quickly talk about the cholangiocarcinoma and the related background, but I have to be honest, I just started to learn this disease, and I don't think I know too much about that. First, I have nothing to disclosure relate with this talk at all. Yeah, and I don't think we need to know too much about that because this is uncommon disease, but getting higher and higher in incidence, especially Asia. And the treatment outcomes still not very favorable, as most of the people are aware of that. Survival rate, of course, is very low. Yeah. And today, I would like to use the next 20 minutes to talk about several points, such as the background, treatment option, and the current molecular mechanism and future direction, which is I believe so. And I start with this slide. I quote from Gauri's talk about the incidence of cholangiocarcinoma. This actually is an increase globally, and start from the incremental increase in the Southern America, and then gradually is Southeast Asia, and now it's quite significant. And if you can see from this worldwide incidence, the green part, that is the higher incidence than the 6 out of 100,000, and the red is about less than 6 to 100,000. By definition of the orphan drug, that's 200,000 cases or lesser. So although we feel that's devastating about this disease, but this also opened one of the great opportunities for us to do the drug discovery about how to apply our new drug in the orphan drug designation, or also consider 505(b)(2) in orphan drug status as well. So there is lots of the workers, the researchers in these fields, and especially in Asia, we need to raise our awareness, and that is all that this conference is about. And treatment algorithm is quite relatively simple. First, it's very similar as the liver cancer. If you have the early stage of a disease, which is stage 1, it's a localized single tumor, you of course go for the resection. If you are stage 2, you have locally the vascular the invasion, and you also go for surgery as you can. But the study confirmed the adjuvant treatment show benefit, so you should consider adding the adjuvant treatment. And the survival in this stage 1 and 2 still can be high, as you can reach about 40% near in 5-year survival. However, once you go for the stage 3 per se, and you will start to see that's a very devastating. You see the visceral organ involvement, local liver function impairment. It's a not surgical resectable. For that, a local treatment can be considered, such as a chemoembolization, radiofrequency. However, that is only added for a few months, and to make this median survival to approximately year to year and a half near. And you need to add chemotherapy, but the chemotherapy itself is also just to carry on the median survival about 1 year. Adding the chemotherapy on the local treatment might show benefit, but that is also carry on some side effect. The chemotherapy, such as gemcitabine and cisplatin, still remain the gold standard. That's almost about 9 years already. And this is the study published in New England Journal of Medicine in year 2010, and that confirmed our new gold standard is the cisplatin and the gemcitabine, which is better than gemcitabine. Of course, the chemotherapy is better than no chemotherapy, which has been proved several years before the 2010. And from there, we start to use the cisplatin gemcitabine as our standard treatment, not just for adjuvant, also neoadjuvant as well. Yeah. And we all kind of agree in this field, adjuvant treatment is necessary, especially for stage 2 and also for stage 3 who finished the local treatment. And for adjuvant, there's only limited drugs. There's cisplatin, gemcitabine, 5-fluorouracil, and lately we added the oxaliplatin into that. FOLFIRI has been tried, but it's also still it's a very hard to accept it. And if we put it all together, consider the surgical is still one of the best the treatment for that, and then follow with the adjuvant treatment, you might push into the 50% the local control. But the question is the not sustainable. And so the first year or year and a half, you might see the response but quickly deteriorate. Oxaliplatin and plus gemcitabine seems like slightly better than cisplatin and gemcitabine. However, the adverse effect, particularly the neuropathy, is the main concern. Capecitabine S1, which is oral drug, an analog from the 5-fluorouracil, also shows some benefit, but this is all the minimum benefit for the few months, the life survival adding. And so this is still very hard to accept it. And for unresectable disease, however, there's so many things you can consider, including we mentioned several times about oxaliplatin, combined alone or with the capecitabine. And in United States, capecitabine. In Asia, we use S1. And sorafenib itself or combination all can be considered. And so this several ongoing trials. Some are completed, some failed. And the last year, people start to look into the Abraxane. After Abraxane combined with the gemcitabine shows successful in pancreatic cancer control. So serendipitously, we consider that into the bile duct cancer. And Abraxane cannot be used in a high dose. And the low dose was recommended, 50 milligram per meter square was the recommended dosage in using in the bile duct cancer. Phase 1 has been confirmed, and the International Cholangiocarcinoma Consortium, as we are here, the slide just to show in the opening remarks that suggest that moving into the phase 2. I think that's very important to see what's that result. It's also open another opportunity using Abraxane and also helping in our patients. But before we talk about the kinase inhibitor, the TKI, I think we all should appreciate the Ming-Chi's contribution in this field, and especially the EGFR, FGFR, etc. And also the main issue is about the pathway rather than just a biomarker itself. And we can see there's several opportunity to look into the different receptor, probably the EGFR, FGFR, are most important, KRAS as well. But recent year, about pretty much as 2 years ago, we start to look into the IDH1, IDH2 mutation because there's a drug, AG120, AG220, and that is all the possible drug now. EZH2 is also, we have a new drug. So these are the new boys on the blocks, and we can start to look into that alone and searching for the dosage, searching for the liver function impairment patient, which is we have, and then determine what's the best dose. And this slide is taken from the Nature Review, and we can look into that all the different drugs and tied in with different mutation. And IDH1 is the one blocked by IDH inhibitor, probably need further study. CDK4, CDK5, control the cycling and the cell cycle is another one. So there's several others, and this is outstanding review, and talk about all the detail in the chemotherapy point of view, the target therapy point of view, immune point of view, and the new drug, the EZH2, HDAC, and the methylation as well, all being discussed and summarized, all the trial, background, and the success or failure rate, etc. So I think this is a very good review, can be referenced. And I like to use one slide, particularly bring up IDH1 mutation, which is we're collaborating with China. And because this is mutated IDH1, even though the percentage is still about just 5% in cholangiocarcinoma, but this is a very unique the biomarker we can follow. If you can identify our patient had mutated IDH1, and the result is that they will increase the 2-HG and the impartially metabolized isocitrate to the alpha KG. So that is causing the further downstream problem, such as the DNA damage and the methylation histone and the cell differentiation changes, etc. So with this, the new drug, we just need to learn how to use that. What is the best dosage, and what is the proper regimen and the scheduling, and that is something we all can learn together. Another drug, which is the FGFR2 from Novartis, BGJ398, and this drug is carrying on the high hope from phase 2, but even to the near phase 3, but apparently they are holding at this moment for lots of the reasons. Side effect is the one of that. But I think the similar drugs will come, or just come from the modified dosage, etc. I think we should all learn together because this drug truly has a potential, not just to block the FGFR2 and also the gene fusion translocation, all can be covered. And so this is a good target in the future. And lots of people is working on the similar drug, which is selectively inhibit the FGFR. In our lab, we also compared to the BGJ398, we developed the compound 145, very, very similar potency, very similar inhibition, and the very similar docking, the binding site. So we are continue working on that. But similar problem as the 398, the toxicity might be a little bit too high, particularly for heart. So I think we need to lower down the dose significantly. And of course, single agent might not be really work enough. Yeah. So this is the summary slide to talk about, to summarize all what we talk about. There's several, the target. There's several drug can be binded in. So with the basket trial, I think that will worth it to look into it. And important is that we all should work together. Otherwise, it's very hard. I'm not going to talk too much about immunotherapy because Ming-Chi already discussed very, very clearly and outlined the future. That we all know the immunotherapy already applied to so many diseases. And the bile duct cancer, which is cholangio, regarded you are testing the Pembro, Nivos, Epi, or combined, all is ongoing trial. Usually it's on the phase 2. But more than that is we have a more immuno-oncology trial ongoing, not just using the immuno-oncology drug alone. We have a immuno-oncology drug combined with the monoclonal antibody, which is one of the potential, for example, the pegylated interferon and the GM-CSF. So this kind of combination has been studied. And another approach is combined with the chemotherapy, which is, again, is usually using 5-fluorouracil, oxaliplatin, cisplatin, or gemcitabine, and all can be considered. And Abraxane, low dose, is another thought. And some people also move on to the combination with the target therapy or cell therapy. The cell therapy can be considered, and they are putting together, but certainly that's a little bit whole 9 yard. But we will need to continue monitor the result and learn together. With all these things is ongoing, and we know we need more. Through this international consortium, I think that can help us to build up the new research and the new tool and new drug discovery, and also find an old drug, particularly for the 505B2 to the new indication. There's a hurdle about cholangiocarcinoma research, because all the cell lines we are using come from different background. Some is we got is from the liver fluke, which is the common background and usually provided by Thai, and that is very helpful. And some are from the gallbladder stone, gallstone, intrahepatic stone. And with all of that, they have inflammation background at the beginning, and then started building up the molecular changes. So the cell line is not very clear. Another thing is it's very hard to just using the cell culture cell line. So currently, people prefer to use the 3D culture, particularly organoids. They mixed with the liver primary cell, which is to make the more reality to the patient's the conditions. Besides that, we use a different animal model or PDX model, and lately try to avoid the model difficulty from the in vivo that we started using CRISPR. But changing one mutated gene is not going to help you. So we try to using CRISPR to transfect all the several mutated genes together, such as using the P10 and a few others to get P53, etc., to do it together. And so with that, there will be more mimicked to the reality. And in our campus, we also collaborate with Dr. Wei Jia in the microbiota. And you can imagine if liver fluke caused the cholangiocarcinoma, if the intrahepatic stone caused the cholangiocarcinoma, that background is what? That background is inflammation. So that inflammation itself is very critical. And the microbiota metabolize the primary to secondary bile acid. And if that is not complete, and then that causes the inflammation. So this inflammation then trigger to the neoplasm. So we are looking at the microbacteria, and in this paper they outline very well, such as the bacteroid, Clostridium, Lactobacillus, etc., all related with a certain biochemical changes, and that is directly associated with the bile duct cancer. And that mechanism in detail can be varied, but important is the build-up ROS, cause inflammation locally, and then trigger cytokine, chemokine, and then start with the transformation and the neoplastic changes. And that is a common agreement about the bile acid and microbiota talk together. In GI cancer, that's also included colon cancer, but particularly because bile acid come from bile duct. So cholangiocarcinoma actually play a very critical role and rely on this this background. So I think this is very critical to look into microbiota and the cholangiocarcinoma. This is my last slide, and I think that we all need to appreciate the consortium, such as Cholangiocarcinoma International Consortium, because that is possible to put these genome, transcriptome, and the cell line exchanges, primary culture, animal model together to make this the whole effort in targeting or just overcome the diseases such as cholangiocarcinoma. We all should contribute whatever we have in the building up together. So this is all my collaborator from Shanghai, and I used to work at City Hall for 30 years, and not 30 years, 20-something years, and before I moved to the Taipei. So we still have collaborator, and we appreciate we have lots of support in this work. And again, this is still a new thing to me, and I'm learning. I wish I can use this opportunity to learn more from you all. And thanks for your attention.
Thank you very much for this very informative talk. Now we open this talk for discussion or question. Professor Hong.
Yeah, you just quickly showed the slide that multiple drugs was there. Then I noticed the PRCA was involved. And then since this disease, one of the standard chemotherapy is cisplatin plus gemcitabine, right? So has PARP inhibitor, which already 3-4 drugs been approved by FDA now, has PARP inhibitor also in the clinical trial? Yeah, it's also there. Okay, thank you.
Yeah. I think PARP being studied in the phase 2 as well. Yeah. But of course, you have to go by all the sequencing and the mutation identification. Yeah, I think you're talking about this slide, I believe, right? Yeah. In this slide, they talk about all the details, like a PARP, EGFR, FGF, and CDK4 and 6, because this is available drug now. But if we want to study these drugs in the not common disease such as cholangiocarcinoma, we have to group. Otherwise, it's not possible. Yeah, you work alone, you can work 10 years. It's not going to help your patient. So I totally support this kind of consortium and build up the momentum and form a team. Yeah.
Any other question or comment? Professor Yan, you.
Yeah, thank you. And if people remember, there is a paper published in Nature about the mutation burden rate. The number one is bile duct cancer. Yeah, you can see that that's a very, very high. Yeah, so we can do a lot of things in immuno-oncology. Yeah, but the dosing, etc., still need to look into in the details. Yeah.
You have mentioned cell line different from Western and the Eastern, especially in Eastern country, liver flu is common. Do you think the treatment modality or treatment should be different between Eastern and the Western cholangiocarcinoma?
That I don't know the answer. Yeah, because you still have to go to the molecular profile to make the decision, because we have a handful of the drugs. If we break down that to the Asian, that might be a little bit hard at this moment. But of course, like Irisa's the story, Asian female non-smoker stand out from the outcome point of view. So it's possible. Just we need to work as a consortium and form a team to look into this special patient demographics.
Thank you. Sure. So based on our experience so far, we have done lots of research together with our collaborator doctors in China. So far, based on over 200 ICC, maybe 400 ECC, and 300 gallbladder, we're seeing some differences for genomic profiling between the US and Chinese population. For example, NTRK, actually, we see nothing from those hundreds of patients by now, unfortunately. But we do see like a HER2 pathway showing much higher, and POP inhibitor-related mutation seems higher than US, and a number of others. And TMB is slightly higher compared US and the Chinese. So lots of interesting things happening. Yeah.
There's a paper published from China in Oncologist, and outlined all the intrahepatic versus extrahepatic versus gallbladder, and their mutation profiling is kind of significantly different. Yeah. So I think that we can work together to look in more details.
Thank you.
Thank you. The final question. The final question.
Talking about cholangiocarcinoma animal model, actually we do have a very simple and useful animal model. We treat the SD rate using chemical induced, and we found that the expression of the molecular profiling between our animal and the human being are very, very similar. So you can check my name, maybe published in recent 2 decades. I think it's a very cheap and useful animal model.
Professor
Thank
Ye
you.
Hassan.
Yeah, thank you. Yeah.